Yeah, thank you, Lisa. Most welcome, and I'm very pleased how many people attend this lecture on, and I will give a little bit broader scope, pharmacotherapy of cerebellar disorders, including CACNAIA 1A. And I'd like to begin with my conflicts of interest. So actually, my name is Michael Strupp. I'm professor of neurology and clinical neurophysiology at the University of Munich. And I have been working with patients with ataxias for more than 20 years. I'm also editor of the Journal of Neurology, and I'm consultant for many companies, and I'm a shareholder, investor, and scientific founder of IntraBio, who is taking care of CACNAIA and acetyl-leucine. We have published quite extensively, and our papers are quite well cited. I would also like to introduce myself by beginning the 4-aminopyridine story. I think most of you are familiar with 4-aminopyridine, this so-called potassium channel blocker. And this will be the first brief part of my presentation. How does it work? In which patient does it work? What can we expect in terms of the mechanism of action and the clinical effects? The second molecule is acetyl-leucine. This molecule is approved in the US by the FDA for Niemann-Pick type C, also an inherited disease, September 24th last year. So these are the different aspects for Fampyra, 4-aminopyridine in episodic ataxia type 2, but also in a symptom which is very frequent in CACNAIA 1A patients, a so-called downbeat nystagmus. That will be my introduction for you. And then we talk about this modified small amino acid in cerebellar ataxias. And I'm also presenting some recent data on patients with CACNAIA 1A. And I think, Lisa, you and the other members of the team can also give their experience to the others with acetyl-leucine. And we will also discuss how you might get approval by the health insurance companies, which is a quite long and painful back and forth pathway, but evidently some have already succeeded. So with all typical German modesty, I'd like to point out that already in the year 2004, I introduced 4-aminopyridine for the treatment of episodic ataxia type 2. This was based on a case series. And later on, we have done a randomized placebo-controlled trial. This is the state of art to look for the efficacy of any molecule. And these were 10 patients with EA2. And you see here the frequency of episodes went down from 13 to about 3. And then in the placebo group, it was about 11. So there was a clear clinical benefit. Another molecule, which has been used since already 1973, is acetazolamide, this carbohydrate inhibitor. And this is also recommended for a symptomatic treatment of episodic ataxia type 2, but there had never been a controlled trial. And therefore, we applied fampiridine, acetazolamide, and placebo in a cohort of 30 patients with EA2. And the major findings were as follows. Both molecules are more effective than placebo. However, another aspect of every treatment are adverse events, side effects. And the number of side effects was much, much higher in patients who take acetazolamide, which you evidently give also your patients with EA2. And this ended up with 24 patients on 4-aminopyridine and only 3 on acetazolamide. So although, of course, not approved by the FDA or the EMA, we recommend
Israel that both of them come to know Jesus Christ.
We recommend Fampyra for the treatment of EA2. And now listen to a patient, because this potassium channel blocker, which, as you will see in a few moments, normalizes the activity of Purkinje cells, has two effects. Listen to this patient and his response to the therapy.
Mr. Rink, you suffer from episodic ataxia type 2. How many attacks at the most did you have per week when we started with the treatment?
Before the treatment, I had attacks about 3 or 4 times a week.
We have been treating you with Fampira for many years, 10 milligrams twice. When was your most recent attack?
Now it's already been roughly 9 months.
Wonderful. Do you also notice an effect on your ataxia between the attacks? Has anything changed there as well?
Yes, I hardly notice the ataxia between the attacks. And my fine motor skills also work really well now.
Thank you very much.
So actually, without this molecule, you would not be able to understand the speech of the patient. So it has a double effect. It reduces significantly the number of episodes, and it improves the ataxia in terms of speech, limb coordination, stance, and gait. And this is something we also demonstrated here in another paper. We looked at so-called gait variability, which is an objective parameter for cerebellar ataxia. And we could clearly demonstrate that patients under the treatment of 4-aminopyridine showed a reduced gait variability, reflecting a positive benefit on gait ataxia. And for many diseases, and you will see more animals, we do have animal models. For EA2, the classic animal model is the so-called tottering mouse. It has a similar mutation, and you see here on the left hand an animal before treatment and on the right hand during treatment. And you can see that the performance is much, much better, less ataxia. The animal on the left can hardly walk. The animal on the right, of course, is not normal, but it moves much better. And now I'd like to show you how 4-aminopyridine works in episodic ataxia type 2. And this is a little bit difficult to explain, but I will try to guide you through. What you see here in the left upper corner is the so-called discharge rate of the key cell in the cerebellum, the Purkinje cells. And you see this is very regular. In animals with the tottering mouse mutation, similar to the EA2 mutation, you see this is irregular. That means an irregular cell cannot be as well as modulated as a cell which fires regularly. And if you apply 4-aminopyridine in therapeutic concentrations, you see that the spike variability decreases. So this cell is transformed into a wild-type normal cell. And this explains the mechanism of action in terms of prevention of episodes and improvement of ataxia between the episodes. I also introduced 4-aminopyridine for the treatment of downbeat nystagmus, which is a very, very frequent symptom in patients with CACNAIA 1A. It's often overlooked because patients are not investigated properly. On the left side, you see a patient before treatment, and on the right side, during treatment. You see the eyes jump much less, the visual acuity is better, postural imbalance is better, and the general performance, functioning, and quality of life is also improved. This is something we also demonstrated with 4-aminopyridine in another placebo-controlled trial. Not only visual acuity is better, but also walking and get up and go test. Now, how do these molecules work? Well, as I told you, the basic problem are Purkinje cells. And if you normalize the function of Purkinje cells, then they release transmitter which inhibits. Then the function here, the interaction in the network within the brainstem, you see here the right side, the cerebellum, the brainstem, and then the inhibition here is normalized. And this can also be directly measured when you have a look, and now you're familiar with these recordings of these core cells. You see here the Purkinje cells, here a control cell, and if you apply 4-aminopyridine, this cell is much more active. If this cell is more active, the release of the key inhibitory transmitter GABA is increased, and this inhibits other cells, and thereby it, for instance, suppresses downbeat nystagmus, improves ataxia, and prevents episodes of ataxia. And we have published quite extensively with 4-aminopyridine, and the major indications are now for the molecule, although not approved, no clinical trials, sufficient or applications by the FDA and EMA. That's the basic reason, by the way. EA2 and downbeat nystagmus. And we have now a broad spectrum of indications. Now we totally switch the topic and come to a small molecule, acetyl-leucine. I will give you some information about the mechanism of action, how it works in cerebellar diseases, namely so-called lysosomal diseases. I'll explain that later. And very recent data in the field of CACNAIA 1A. And finally, we can discuss some perspectives. And this is a statement required by the FDA. So Agnersa is an investigational therapy that has not received FDA or any other regulatory authority approval and has not been demonstrated safe or effective for any use except for Niemann-Pick type C only at the moment. And these are the topics we are talking about. You know a lot of amino acids. Acetyl-leucine is something very special, and I'll tell you why it's so special. In the second part, I'll briefly explain how it works and how it may also work in CACNAIA 1A diseases. Then as an example, and this can be an example also for a trial in CACNAIA 1A diseases, I'll show you the trial design and the results of a so-called phase 3 placebo-controlled trial, which we published last year in the New England Journal, and which was the basis for the approval by the FDA. We briefly mention other diseases, and finally, acetyl-L-leucine in CACNAIA 1A disease. This is a summary of the history of this molecule. So 2013, I introduced it for cerebellar diseases in a case series. Then we did animal studies in animal models in Oxford. This is the trial I mentioned, and this can be exemplary also for CACNAIA 1A in the New England Journal, approval by the FDA and mechanism of action. We have published also very extensively on this small molecule, and luckily, the molecule was declared as the drug of the month March 2025 by Trends in Pharmacological Sciences. So now we dive a little bit deeper, mechanism of action, and what can we expect from this molecule in various diseases. But before we move on, as I said, you're familiar with amino acids. Amino acids are the essential parts of any protein. What is so special about leucine? Leucine is an essential amino acid. That means you have to take it from outside by food. Leucine is the only amino acid which is not metabolized in the liver. And we talk not only about leucine, but a chemical modification, so-called acetylation of leucine. That means there's another group with the amino acid. And the effect is tremendous because this modified amino acid is transported through all cell membranes by a different transporter, which has a very high capacity, and that's essential. So at the end, every cell in the body has a higher concentration of leucine, and we demonstrated that leucine inside the cell has a major impact on the function of cells. For instance, cells have more energy, and the general gasoline of cells is so-called ATP, adenosine triphosphate. And normally you produce a certain amount of this ATP per glucose, and in cells which do not work properly, ATP production is reduced. If we add leucine, the cells produce more energy and are in a better state. And It was also demonstrated that it normalizes the membrane potential of cells, which is essential for the electrophysiological functioning. And here, this and the next 2 slides show you how it really works. So N-acetyl-leucine, it goes through all membranes and it reaches the cell, namely nerve cells, namely cells in the cerebellum, and it improves the function of cerebellar neurons. Here's a little bit of biochemistry on the left side. Well, this summarizes what I described before. You produce more ATP, every cell has more gasoline. Cells produce less lactate, but they produce more ATP, and this has an impact on many, many mechanisms inside the cells. And this is a paper which we just published. It shows that the effect is dose-dependent, concentration-dependent, and one of the key players in the cells are mitochondria and lysosomes, and they are essential for a proper functioning of every cell. In other words, you see all the details, and we know all the details on how this small molecule works. This here shows, and this may be familiar to you already, recordings. You see here so-called action potentials. And what acetyl-L-leucine is doing, it normalizes the excitability of cells. And this is crucial for Purkinje cells. And this is crucial for the effects in CACNAIA 1A diseases. Now you see another patient with ataxia. That was actually the first one we treated. This is a standard test. You're all familiar with this test, maybe in your children, the 9-hole pegboard test. And on the left side, you see this boy for treatment. And on the right side, you see his performance during treatment. So you see his functioning is much, much better. And this has impact on quality of life and functioning in daily living. These are the results of the first case series, which we published in patients with different types of ataxias. And now we shift a little bit to a disease which typically shows such an impairment of eye movements. You see, this young girl is not able to look up and downward properly. If you see that, it's very likely that she suffers from a genetic disease, Niemann-Pick type C. And in Niemann-Pick type C, and that was another cohort, we tested acetyl-leucine. We saw a clear benefit. And you're now familiar with this test. Left before treatment, right during treatment. You see this girl is much, much better with acetyl-leucine. And this is the trial design, and I will guide you through that. And this can be exemplary also for CACNAIA 1A of the study which we published in the New England Journal last year. So patients are either on the drug for 12 weeks or for 12 weeks on placebo, and then they switch. And at the end of each period, we determine the performance of the patient. Another thing which we have added to all trials is the so-called extension phase. We look at the long-term effects over 12 and 24 months to look at potential neuroprotective effects. Here a summary of the results. We used the so-called scale for the assessment and rating of ataxia, and we saw a change from baseline of almost 2 points in the treatment group. And here you see a patient, and you see the difference, left with a rollator, and right, he can walk unaided. So this has an impact on functioning and quality of life. Another aspect of alveolar molecule is, of course, safety. And here you see a summary of the results. On the left side, the treatment group. On the right side, the placebo group, 59 patients. So there was no serious adverse reaction, and the frequency of adverse events was similar in the treatment and the placebo group, and the side effect was very minor. So null is very well tolerated. And these are the results which I'm going to present upcoming Monday at a meeting in Helsinki. And let me guide you through that. Here you see the timeline. up to 24 months. These are patients with Niemann-Pick type C. In blue, you see the change over time in the treatment group. And here, as in many diseases, there's a natural history group. And the natural history group, you see that these patients with NPC steadily decrease. And here, under the treatment of acetyl-L-leucine, after 24 months, they're even better than at the beginning of the treatment. So this indicates a neuroprotective disease-modifying effect, which is, in addition to the symptomatic effect, essential. Now let's have a look at another child. On the left side, Dario. He has a different genetic disease before treatment, and on the right side, during treatment after 6 weeks. So you see the clear difference in this patient with a genetic disease leading to ataxia. And this was also reflected by another clinical study. The disease is GM2 Tay-Sachs-Santoff disease. Here, during treatment, a clear improvement, washout period deterioration. And you're familiar now with animal models, and this is now an animal model from Oxford. Here you see the untreated mouse and the treated mouse. This is like a race between Oxford and Cambridge, and the Oxford, that means the treated mouse, is much better. And for the other disease, there's also an animal model. This is very helpful to look at the mechanism of action and the clinical benefit in various diseases. And of course, there are also animal models, as you saw, for CACNAIA 1A. And one, of course, can also test the mechanism of action and the benefit of this molecule in these CACNAIA 1A models. The animals treated live longer than those not treated, disease-modifying effect. So we see in animals, in men and mice, the same effect on disease modification. Now, at the end, we switch over to CACNAIA 1A diseases. I think you're all familiar with the clinical manifestations: EA2, familial hemiplegic migraine, progressive ataxia, and epileptic seizures. Let's start with epileptic seizures and fampredine. I think the risk is very low that you really induce in the dosages which you give epileptic seizures, but nevertheless, we have to be careful. Now I come to case descriptions and case series. CACNAIA 1A mutations. This is something we published in 2020. So there was a patient with a new mutation, and they responded very nicely to a treatment with acetyl-leucine. Here, another case series from colleagues published in the European Journal of Pediatric Neurology. This year, they had 4 patients, age 15, 5, 9, and 14. They received acetyl-leucine. The effect was evaluated by the scale for the assessment and rating of ataxia and clinical global impression. In all children, there was a rapid improvement of ataxia. The SARA changed by 3 points, which is clinically highly meaningful, and the improvement was sustained for up to 3 years. So these case series show that N-acetyl-leucine can be effective in CACNAIA 1A patients. So summary: 1 published single case, 4 cases published with a monotherapy. We can now talk also about your experience with acetyl-leucine. And there's also a summary of cases by the CACNAIA 1A Foundation. And another thing is, for the future, this can be a trial design, genetically proven CACNAIA 1A diseases. It's a repurposing of a drug, which is now approved for NPC in the US. The dosage will be the same. Treatment and observational period depend on what we are looking for. If we look at, for instance, ataxia episodes, a treatment period of 12 weeks will be sufficient. If we look at disease-modifying effects, the treatment period should be at least 12 months. Of course, such a trial will be placebo-controlled, and we have established endpoints. And of course, the study protocol has first to be evaluated by the FDA. So this is the final slide of my presentation. So we talked about Vampira in episodic ataxia type 2. Most of you are very familiar with that. Acetyl-leucine, some of you are familiar with that based on individual cases. We talked a little bit about the perspectives, and questions preferably should be asked by doctors because of regulatory issues with the FDA. But this is not so super strict. So I'd like to stop now my presentation here, and Lisa will take over and run the show. Thank you for your attention.